Good afternoon, everyone. I just want to thank everyone for joining us today. For those of you who were not able to make yesterday's session, welcome. We've got a great lineup of 9 companies. I am going to turn it over to Mike Szymanski from the AngelMD team and let him give you a little more context about the day.
Great. Thanks a lot, Jen. Much appreciated. So the Alpha Summit came about because of our need to integrate all of the educational assets and mentorship and connections that we have as individual supporters of startups when they're going out there looking for success. But one of the things that also came about was the need to create kind of a critical mass of great talent and great startups in order to prove to the investor base that this is a space that they should be pushing more and more of their attention. I really am sick of watching SPACs go up and having bad companies be able to raise $500 million on the secondary markets when these amazing startups are having difficulty filling out their Series A or Series B rounds. So part of the Alpha Partners is about that. Today, we have the amazing opportunity to watch 9 of these companies present from the best of the best. So Dr. Richardson and I have had the privilege of working with most of these companies over this past year with Pitch Club, and a lot of them have come through that event as alumni. But they're also alumni of other great incubators and startups. And each of these companies has a story to tell in terms of people that helped them when they were starting up, invested in them, gave them practical help from their mentor base or educational help as they went along. And so we're going to introduce each of these companies with a brief 30-second intro from their sponsor, or I will do the introduction in the name of their sponsor if they weren't able to make it today. And we will show you just what kind of breadth and depth is available in the healthcare startup space. So with that, I'm going to launch off with Vivek Kumar and Nangiotex. Now, Nangiotex was supported by, Vivek, who would you say was your biggest supporter?
Well, right now would be the NIH.
The NIH.
SBIR grant. Yeah.
Critically, you guys got your one of your kickoffs from the Mass Challenge, winning the Mass Challenge Boston event, correct?
So yeah, we took part in that, yeah.
Great. So we'll lump the NIH, but NIH hopefully is supporting everybody. Let's single out Mass Challenge as one of those innovation events that really gives a lot of these startups their help. Nangiotex is a remarkable product rebuilding tooth pulp in otherwise dead teeth. And with that, I hand it off to you, Vivek. Please take it away.
So hi, everybody. My name is Vivek Kumar, and I am the co-founder of Nangiotex, Inc. Our company is focused on regenerating blood vessels. Our initial go-to-market strategy, our initial product, is a regenerative hydrogel that can regenerate dental pulp. We know that root canals are painful, caused by infection in the tooth. An endodontist, a dentist, goes in, removes the infected pulp, and the current standard of care is putting in rubber rods known as gutta percha that leaves the tooth, especially juveniles, mature teeth, that leaves the teeth unable to regenerate, prone to fracture, and ultimately requiring a restoration. So what we've done at Nangiotex is develop a material, an off-the-shelf material that can regenerate dental pulp. This hydrogel is based on an invention that I and my partners came up with a couple of years ago, and we've established this platform to regenerate robust vasculature in a number of different animal models. Most recently, I think 3 or 4 months ago, we published a study where we regenerated dental pulp in a canine model, in an adult large animal model. We showed robust regeneration of vasculature. For any of the biologists out there, all of these little red circles are blood vessels that are lined by endothelial cells and smooth muscle cells. They're mature blood vessels that populate with dental pulp stem cells within the pulp space within just 30 days. We measured this against a number of different controls and showed that our regenerative hydrogel performs significantly better than the standard of care. Having established the efficacy for this biomaterial in dental pulp regeneration and having established the platform with millions in funding, and most recently, as I mentioned, an NIH phase 1 SBIR grant, we're ready to take this towards your key pivotal CMC ADMET studies and phase 1 clinical trials. For this, we're going out to raise approximately $3 million that will give us a read on whether this works in a patient population. We envision phase 1 clinical trials to include patients who require root canal, right? doesn't really make sense to do a safety study and say, hey, you want to get a root canal for fun? Anyway, we plan on recruiting a patient-specific population, getting an efficacy readout in phase 1 itself for a modest investment. We think we can take this to market within the next, or an exit within the next 18 to 24 months, demonstrating human efficacy, and then either partnering up with a strategic or large pharma, or doing a subsequent raise to take this through phase 2 and phase 3 clinical trials for approval of this drug. Notably, this is approval of a drug in the pharmaceutical space. In addition to that, the dental and craniofacial space. So the barrier to entry is lower, but at the same time rigorous. And we think that this platform initially will target root canals. We know it's $1 billion market, a $5 billion global market with a CAGR around 6%. Based on current reimbursements, you go to the dentist, they pay about $100 for the consumables. Even with counter reimbursements, because our cost of manufacturing is about $1 per dose, fill and finish at scale, we think we can make significant profits even with current reimbursement structures. We have a stellar board. I'd like to think the technology is pretty awesome. That was invented by the folks that you see on the screen here in the top left. We have key strategic support and advice from our Chief Scientific Officer, Karel Petrak, who has experience taking these drugs to market. In addition to that, we have clinicians who specialize in pediatric dentistry, our initial target market, as well as board members of the American Association of Endodontics, Craig Hirschberg, couldn't be here today. We have Mike Bowie to guide us through regulatory advancement, as well as I'm sure some of you may know, many of you may know Jeremy Krell, who's a phenom when it comes to business development in this space. And of course, our basis in vascular tissue regeneration with Elliot over at Harvard Medical School. Thank you very much for the opportunity. I'm happy to take questions. Please feel free to reach out. Our website is down there, nangiotex.com, as well as my email address. That's my first name, Vivek, at nangiotex.com. Thank you.
Vivek, thank you so much for being here today and for sharing your company with us. Really cool technology. You know, it's been a while since I've seen this presentation, but it again wows me even the second time through. So for those of you that are in the audience, we would love for you to put some questions for Vivek in the Q&A. But I'm going to start with one of my own. So all of your studies to date have been, sounds like, in animals. So what hurdles do you see in transitioning that into humans? And specifically, how do we know that the pulp that's regenerated is supportive of long-term health?
Right. So one of the studies that we didn't do in these canines was ask them, how does it feel? And does it hurt, right? And unfortunately, that's very hard to do in animal studies. One can do pulp sensitivity testing and other efficacy measures, which the FDA, the primary endpoint for the FDA is what you see on the screen, robust regeneration of tissue, vascularization. In addition to that, we've seen nerve regeneration. We plan on doing longer term efficacy studies. It's not required by the FDA, but it's part of the package that we're planning to do. Coming back to your question, how do we know? We've done a lot of studies in animals. That's true. We've done mice, rats, dogs. We've injected this in numerous different tissue spaces. What that has given us is a sense that this is a safe material in high doses. It is safe in multiple different tissue spaces. It biodegrades and is replaced by native tissue. It doesn't cause an immunogenic response. We've dosed it multiple times. So what we need to do in reality is fill in the gaps. Right? Every drug that goes to market needs a general series of GMP, GLP tox studies. You have to contract that out to CROs. That's what we're raising funds for. We've de-risked the technology. We think the next pivotal inflection point is human trials, and that's what we're raising for.
Great. You know, the other thing that I would wonder about is how is this going to change clinical practice?
Right. So right now, a clinician goes into the tooth, removes the infected pulp, disinfects the pulp by putting in different solutions, rinses the pulp with saline, and then carefully places these rods, cuts these rods, as you can see on the right, until it's fully packed and then caps the tooth. We're going to simplify it. Those rinses I mentioned just now, that last rinse inject a hydrogel. These hydrogels, as I mentioned before, flow like water. Inject it into the tooth space and cap the tooth. We will reduce procedural time, we will reduce procedural cost, and we will easily slot into current reimbursement strategies. So that's why we're very excited about this technology. We reached out to key opinion leaders in the space, Henry Schein, Dentsply, these key leaders who supply gutta-percha. And the future of dentistry, at least endodontics, is regeneration and maintenance of the pulp. No longer are we going to cap it and leave it there, hoping it doesn't crack or chip or fall out. What we're trying to do now is to regenerate vital tissue within the tooth.
That's just incredible. We have some questions from the audience. Can you talk about your IP?
Definitely. So the IP behind this technology, I have a slide somewhere here. It was filed at Rice University, where I did my postdoctoral work. My professor back then was Jeff Hartgerink. We've maintained an amazing relationship. We spun out this company together, and we've continued developing this platform. We've filed subsequent IP on looking at other aspects of this platform. I'm happy to go into more details, but it's a fully granted patent. It was granted in 2016, I believe. We think that we'll be able to get new chemical entity status because nothing like this, a self-assembling peptide, has been on the market. So it's a new chemical entity. In essence, we think we'll get another 3 to 5 years of market exclusivity granted by the FDA after the patent is up.
Great. And another question, just to take your explanation of how this all works. So you showed us that there's revascularization that you've seen in the tooth tissue. How does that then, those new vessels kind of reconnect to, again, where that supply is coming from, which is in the jaw, correct?
Right. So end of the day, there's no magic, right? So we think that this material is magical or has regenerative properties, but the body does too. When you get hurt, when you get a cut, what happens is you get a fibrin clot, the body's own scaffold that infiltrates with cells, and you build tissue, scar tissue, what have you. When it comes to a tooth, your body doesn't do that well. When it comes to many organs, actually, your body doesn't do that well. It doesn't have enough factors to stimulate cells to grow in, to stimulate responses. You might hear there are many technologies that use stem cells. The problem with the stem cells is that majority of the cells you put in die the minute you take it out of the bone marrow and put it in the lung or somewhere else. When you transplant anything, there is mortality. What we're doing here is taking the salient factors that these cells need, a pliable matrix to infiltrate into, VEGF signaling, vascular endothelial growth factor, very specific signaling to encourage growth of existing vasculature into the tissue. We're capitalizing on the body's own repair mechanisms and providing a scaffold for it to do that, for it to regenerate.
That is incredible. And one last question for you, just because as I think about this, and you hinted at it, I think when you were talking about your team, I am guessing this drug could be applied to other circumstances or use cases outside of dentistry. Can you tell us a little bit about that?
So it's interesting. Our company started, we went to Mass Challenge. We did a lot of different things focusing on peripheral vascular disease, poor circulation in the legs and arms. You're overweight, you have poor circulation, and you get wounds and the tissue dies, it becomes necrotic. One of the issues with peripheral artery disease is that so many different people have tried similar strategies and there's too much risk. In addition to that, clinical trials are kind of expensive. Your endpoints for phase 2 clinical trials take several years. You're looking at limb salvage, whether someone keeps their leg or not. For dental pulp regeneration, another focus of ours, I wouldn't call it the low-hanging fruit, but it's our path into the clinic from which we can expand. For dental pulp regeneration, we're looking at 3 to 6 month endpoints, which are radiographic, relatively low cost for clinical trials, as well as getting a human trial for this platform that we can exit for more, summarized in this slide here. Peripheral artery disease, wound healing, as well as dental pulp regeneration.
Fantastic. Well, thank you so much for being here today. We really appreciate it. And thanks to our audience for helping with the questions today. We're going to move on to our second presenter, who is William Altman with Coronova. And to introduce William, we have Jess Mabus, who was a supporter of Coronova.
Hello, good afternoon, everyone. My name is Jess Mabus. I am a managing director at Nationally Ranked Accelerator Generator. Coronova graduated from our G-Beta MedTech program in 2018. They beat out medical and healthcare startups from across the world for one of these 5 exclusive spots in our program. Our corporate partners were so impressed. This included Boston Scientific. They saw so much potential in Coronova's cardiac assist technology and recognized its potential to be a game changer in the field of heart failure. I would like to introduce William Altman to the stage, and he will tell you more about this revolutionary technology.
Great, thank you.
Thank you very much. All right, well, thank you. Yes, Coronova is developing a really breakthrough cardiac assist device for heart failure. And we've been supported by $6 million in funding from Wellcome Trust, the world's second largest medical foundation after the Gates Foundation as one of our prime supporters. So we're very excited. Also have generator support in that. They've been a great platform, introduces all the players in Minneapolis and many others in the country and in medical devices. So it's fantastic to have their support. And thank you, Jess, for being here today. We also are named by Angel Capital Association as having the best investment potential among the top 3 in 2019. And we'll talk later. We've had many other awards, which you can see on our website. We're dealing with the unmet need to treat acute heart failure. That is a short-term episode in which the heart fails to pump enough blood to support the body. And those, there are existing devices, but the existing devices that support this type of thing to give you time to bridge to recovery, they're limited by extensive blood device interface, which causes adverse events. Here's an intra-aortic balloon pump, commonly used, not much impact. This is a percutaneous axial flow pump. Abiomed's Impella device sells for $25,000 each, but you can see the huge amount of blood contact and churning. And this is the only Bioventricular device which is external to the body with a huge amount of blood contact. And that's very problematic because blood contact is extremely bad for you. 2 to 15% rate of stroke arises in use of these devices over only 5 days. Some stroke can kill you. You have 5 to 13% rate of red blood cell destruction, needing transfusions, and even have 21 to 47% rate of kidney dysfunction and 12% outright kidney failure with VA ECMO. So that's problematic. And in addition, 40% of patients can't use existing devices, leaving a large unmet need for a device like ours. People have calcified arteries. They can't put these devices in their femoral arteries. Others can't use anticoagulants, which are required to prevent stroke. And others need both ventricles supported, and existing devices only treat one ventricle. Our solution is this thin film polyurethane cup. It goes around the heart. It has a nitinol wire framework to make it collapsible. There's no blood contact. We provide biventricular assist. It's rapidly implanted and provides a very gentle, direct cardiac compression. Hope you can see this video. This shows how it can be folded down into a 1-inch delivery tube, make this minimally invasive incision between 2 left side ribs. We then insert it into the bottom of the pericardial sac, their 1-inch incision. It's inside the sac. We have inner chambers we fill with saline to conform it to the heart shape. And then we have outer chambers that we fill with air in synchrony with the heartbeat to gently squeeze the heart, just like a surgeon would squeeze the heart of a patient in distress on the operating table. We can do that 144,000 times a day with our external pneumatic driver, which sits by the bedside. This shows in a large animal model and a sheep how you can see how quickly and easily this device is implanted around the heart. Sheep heart's about the same size as a human. There's no direct attachment. It's super easy to put in. We've had a 100% success rate with over 55 deployments by 6 different surgeons and 23 one-day large animal studies. It's estimated 30 minutes to device activation. Comparable other devices, and really seen as less invasive than minimally invasive aortic valve replacement, which was done in people in their 80s. The device works by enhancing cardiac motion. You can see the device off on the left and then turned on the right. And that gentle squeezing, very safe to do, increases output by 50%, which is enough to mean the difference between life and death for these patients. So a very simple concept, hard for others were not able to execute it, but our founders perfected this device and many improvements which allowed us to do this. This just shows some of our published preclinical animal data that shows the device on. You can see in the center of the slide, increases aortic pressure significantly and increases aortic flow. a liter a minute or 50% because it's increasing the flow out of the left ventricle. For example, this device, a point of entry for us could perfectly be better biventricular support. The only approved short-term biventricular device is via ECMO, has 15% rate of stroke, 20% rate of bleeding, complications. We wouldn't expect to see any of that. And we've even had surgeons reach out to us and say, gee, I wish your product was on the market because we need something for these patients. Our pathway to market is through the FDA. We plan to begin first in human in 24 months, complete in 36. Currently raising $2 million to finish our 14-day large animal studies, between a major inflection point and then proceeding with an institutional round, with retaining an investment banker to get us to begin first in human and complete within 36. And that's a major value inflection point with other PMA device companies in the cardiac space exiting for over $400 million in cash. So we're very excited about that. We have a fantastic team, including Boris Lashinsky, who invented another LVAD that sold for $500 million. Susan Alpert, former chief regulatory officer, Medtronic, is an investor, board member of the company. Dr. George Letsu, Texas Heart Institute. We've had many of the top thought leaders in heart failure join our team. We're very excited to have their support. So we've got a breakthrough technology with great animal data. There's high reimbursement in place. There's early exit potential to premium first in human data, and we'd love to talk to people about it that would like to learn more. Thank you.
Great, William, thank you for being here. This is a fascinating device, and it seems like it has not only a lot of potential, but a lot of use cases. So can you talk a little bit more about what those clinical use cases would be? Is it more of a short-term device, a longer-term device?
No, that's a very good question. No, it is a platform technology that could do both, but we are starting with short-term use. Less than 7 days is the approval that we're targeting. The existing marketplace for devices that are approved for less than, say, 5 days is about $1 billion in current sales. And about 40% of patients have no device at all. So we believe that we could create add to the existing sales by at least up to 400 million. And it's a huge market. Abiomed is the market leader with these percutaneous devices used for less than 5 days. And they have 800 million in sales and a 16 billion market cap. So short term's a huge market, but much cheaper to access or more cost efficient for investors because clinical trials are short. We're only going to use it for 7 days, so it limits our length of our clinical trials and allows us to move to market much faster than if we did a long-term use. But longer-term use would be definitely what I think one of our strategic acquirers would do with our technology, that after doing this first in human, they would seek to not only do it for short-term market, but for medium and longer-term uses.
Great. So in these cases, again, so the patient has this device inserted for 7 days. Couple of questions. What prevents it from being dislodged, and is it easy to remove, as easy as it looked like to insert? And then what next for the patient, usually in these cases, after those 7 days?
Sure. It stays inside the pericardial sac due to the fact all free air is removed from the chest. It's like a suction attachment and like a balloon and a cup of water. If you pick up the balloon, the cup comes with it if there's no air around the balloon. It does come out very easily. I have another video I could show anyone interested, showing it coming out as easily as it goes in, but you do have to give it you have to open the chest, allow some air to get into the chest, and you have to give it a gentle tug to break the seal around the heart. So it stays in place. We found that generally that only a sharp pull on it would be necessary in order to dislodge it. After the 7 days, we anticipate these patients would be bridged to either recovery to go home, or in many of these patients that might be decompensated heart failure patients have a comorbidity like a lung problem or kidney problem. And so they're knocked down with that. They need to fix that lung or kidney problem. And once that's fixed and increased output can show that can recover the patient, then they would become eligible for a long-term LVAD or for a heart transplant, or be able to go home and wait and do that later. So we would anticipate initial indication bridge to decision or bridge to recovery.
Great. That's helpful just to understand how it fits into the workflow, I think, a little bit. One other question I would have is what type of monitoring is required during that 7-day period? And then just from a clinical perspective, again, how much of an advantage is that biventricular support compared to what's on the market now?
Sure. In terms of monitoring, I mean, we'll have an external pneumatic driver, just like a balloon pump. We monitor pressures within the device. We monitor, we look at the ECG signal because we trigger off the ECG, we follow the heart. But it's much, because it's such a less invasive device, I mean, you need a full-time perfusionist monitoring an ECMO device 24 hours in the hospital, and ours should be much simpler to monitor because we don't have the type of adverse events that can happen so quickly with ECMO. And let's see, I'm sorry, the second half of your question?
Was around the advantages of the biventricular support.
Right. Yeah, no, we've really had a lot of clinicians tell us this is really important because right now the only biventricular device is really that external VA ECMO to the body, and it has that 15% rate of stroke in 5 days. So there's many patients that really need biventricular support, but they're reluctant to give it to them because of the huge adverse event rate of that device. So it should really expand the number of patients who get help and really help that population with biventricular support. And we think that's as much as 20% of the market.
Fantastic. Well, thank you again for being here today. And we look forward to hearing about your continuous successes. I definitely think even since the last time we had you presenting on Pitch Club, you guys have gained a lot of ground. So congratulations.
Thank you very much. Yeah, we've just finished. We're in the midst of 14 large animal studies. The data is looking really good. So it's exciting.
Very good. Thanks for being here today.
Thank you.
We're going to move on to our next presenter, and that is Dr. Reid Waldman with Veradermics. And Reid is going to be introduced by Musan. I hope I'm pronouncing that correctly. And we look forward to hearing your introduction.
Hey, thank you, Katie. My name is Rashawn Durek. Glad to be here. I'm an investment portfolio manager at Worth Family Investment Associates. 2 days ago, we actually closed our investment round in Veradermics, and I'm happy to open it up to Reid Waldman, co-founder and chief operating officer of Veradermics.
Well, as you guys said, I'm Reid Waldman. I'm a dermatologist by training and one of the co-founders of Veradermics. And I'm really excited to tell you all about how we're changing the way that pediatric warts are treated. And, warts are incredibly common. Something like 20% of school-age children have warts, and up to 2 million children actually go to the doctor for treatment of their warts every year. And even though this is an incredibly common problem that's bothersome, there are no FDA-approved prescription treatments for warts. And the treatments that are used, like freezing with liquid nitrogen, are either scary, painful, or require daily applications for months. And as a result, the scene shown here is something that's familiar to me, familiar to Katie in our daily clinical practice, which is, parents and the medical assistant holding down a screaming child as we do a painful procedure that the child doesn't want done to them. And, to make matters worse, even when we do these painful and scary procedures, the response rates aren't great, with failure about half the time. The good news is that there's an emerging treatment that works better than any other, and that's injectable immunotherapy. And the idea here is that a foreign protein is injected into the wart to activate the body's own immune system to recognize and fight off the wart virus. And while this is an incredibly efficacious treatment paradigm, the issue is that this injection with the needle is exquisitely painful, about a 7.8 out of 10 on the pain scale. And so as a result, we decided to take this, known effective FDA-approved ingredient and incorporate it into a painless, kid-friendly drug delivery platform, which is a dissolvable microneedle patch that we call the wart patch. Now, the wart patch is applied to a wart like a Band-Aid in a process that is virtually painless. Once applied, the drug-containing needles dissolve like icicles in the skin, releasing those known FDA-approved immune stimulants. The patch is actually only worn for about a minute, and after that, it can be removed, discarded, and you're done for another 2 to 4 weeks, which would be repeated until the warts are gone. Now, at this point, we've made major advancements in the development of microneedle technology and have actually been able to formulate microneedles that contain the known dose, that we want to incorporate into them that dissolve with the dissolution characteristics that we like, and that are strong enough to penetrate through human skin, which is a thick, or through pig skin, which is a thick human skin analog. And we actually have placebo patches that have been able to penetrate through human skin. So where we're at right now from a formulation standpoint is that we have several candidate formulations that we've shown have this stability and dissolution characteristic. We're now casting them all into microneedles, putting them in longer stability studies, and selecting a formulation that can be advanced into cGMP development and then progress into a clinical trial. From a regulatory standpoint, this product is regulated as a biologic-led combination product. And so far, we have had interaction with the FDA via an Interact meeting request that just took place. And our next step is for a pre-IND meeting that would be at the beginning of 2022. And there are really two important things to know from a regulatory standpoint about this product. The first of which is that there's precedent in the wart space that we can run our clinical trials in children and adults simultaneously, meaning we would get FDA approval in both age groups. The second is because this is a first licensure biologic, we actually receive about 12 and a half years of regulatory exclusivity upon approval, which is really helpful. Now, in addition to that regulatory advantage, we also have a fairly broad intellectual property portfolio, some of which is company IP that's been filed around the idea of actually delivering these immune stimulants into the skin to treat skin conditions, as well as methods of use and preparation surrounding our specific formulations. Importantly, though, we've also been in a licensing agreement with a leading microneedle developer where we licensed this very broad IP portfolio that has provided us with freedom to operate. And you don't have to, take my word for it on the FTO standpoint. As part of our Series A financing that Mushan mentioned, we actually had an FTO search performed. And, what was identified in that is that no issued patents, posed a clear barrier to our freedom to operate. Now, in addition to warts being a huge unmet clinical need, there's significant financial potential here simply because of the sheer number of patients that go to the doctor for this. Now, there are about 14 million individuals who actually have warts in the US at any given time, 5 to 7 million of whom go to the doctor. And then the ones we're most interested in are those 2 million who are children. Now, what is important about this product is the same dose of the same active ingredient in the same drug delivery platform can also be used for other indications such as molluscum, which is a viral skin infection in kids, certain types of non-melanoma skin cancer, and genital warts. Now, in addition to the wart product, we have expanded our pipeline into 2 other assets that are currently undergoing development. And as Mushan said, we really urge you to check out the article from earlier this week in Forbes about our Series A, where we closed on $20.7 million to finance our program through into phase 2, and very thankful to the investors shown on the right who helped us there. Importantly, we're really happy with where these programs are at. We're very excited to get this in the clinic. And if you're interested in potentially, starting the conversation about our next financing round, or think that there's a partnership to be had, would love to speak to you. Thank you very much for your time today.
Reed, thanks for coming back and giving us a glimpse into your company today. Exciting news about closing your funding round. That's very cool. Congratulations.
Thank you. Thank you. I will tell you, that was a long, arduous process, and I am very glad that it is over. As the last couple of weeks, as Mushan can attest, there was a whirlwind of just little fires everywhere that we put out to finally get things done. But, really happy and very thankful for all the investor support here.
Great. I have a few questions, and hopefully our audience will put some in the Q&A as well. Why do you think others haven't done this already? Clearly, warts, are a big problem, as you say, and you're right. Oh my gosh, the treatment is very painful for the most part. So why hasn't somebody else looked into doing this?
So I think there are 2 components to that. I think one is, why are there no FDA-approved prescription treatments for warts? And I think the answer to that is a little complex, in that people have tried. If you look in recent years, Claris Pharmaceuticals, which is really rebranded as a rheumatology company, they had a product that made it all the way through phase 3 trials and then just didn't demonstrate efficacy. Verica Pharmaceuticals that just had a CRL rejection notice. They also had a product that went to phase 2 trials for warts, similarly had issues. And then Novan Pharmaceuticals, another company, had phase 3 trials in molluscum, which is an adjacent space. So I think the interest has definitely been there. I think it's just that the execution to date, hasn't necessarily been what is needed. As far as why is no one using a microneedle, I think it's really a timing issue. the group that we licensed IP from invented the microneedle back in 1998. And, really the maturity of that technology to be ready for prime time, ready to enter clinical trials did not exist until the last, year or two even. And so I think it's really more a timing issue than anything. The active ingredient we've used has been around for more than 70 years. But again, without the right drug delivery platform, really hard to make a case to use it.
Yeah, and you talked about in your presentation, the molluscum, the skin cancer, some other dermatologic diseases that this might be used for, but I'm guessing there's a lot of uses for this technology outside of dermatology as well. Can you talk a little bit about that?
So our company specifically focuses in the dermatology space. And if you saw our big rebrand, the idea now that we're kind of moving beyond the single microneedle product is that we're dermatologists, using end user knowledge to develop products for dermatology. But certainly microneedle technology broadly has, significant potential. I am a firm believer that in the next 5 years, every single one of our vaccines will be a microneedle, and the idea of getting a shot will feel very antiquated. Well, that's not something that Veradermics specifically has interest in developing in. The Gates Foundation, other organizations certainly do. And I think that we'll see that technology mature most there.
Fantastic. We do have a question from our audience. Is there a use case for this potentially in cases of alopecia?
So yes, I think it depends which type of alopecia you're talking about, right? The probably biggest alopecia market would be either androgenetic alopecia in men or female pattern hair loss in women. And I think the difficulty there when we think about microneedle technology is, even though there's certainly a drug delivery issue, right? That the reason that Rogaine is not a super effective treatment for that, even though we know the active ingredient grows hair, is because the skin on the scalp is so thick. The problem with a microneedle for a scalp-based approach would primarily be that, there's so much surface area, right? And our specific microneedle patch is like less than 5 millimeters. It would be very hard, I think, to have one go on the entirety of the scalp. But thoughts are there. I think that our company has other interests within the alopecia space, given the size of the market, given the degree of unmet need. But microneedle technology itself, definitely it's been, I think someone patented, minoxidil in a microneedle patch. But I think that there are logistical issues that would prevent that from ever, becoming a product of commerce.
Great. And another question from our audience, would this be used as first-line treatment? And how do you see insurance companies viewing reimbursement compared to existing treatments?
So I think I'll cover reimbursement first and then go into why I think that lends to this being a first-line treatment. There is a really strong, example of how insurers view warts, because almost 2/3 of warts are treated with liquid nitrogen cryotherapy. And there's a very specific CPT or procedure code that is reimbursed for destruction of a wart with liquid nitrogen. And that reimburses from Medicare somewhere about $130. So that kind of sets the baseline as to where wart reimbursement should come in. And because of that and the lack of prescription products, we plan on using a competitive pricing model to say, hey, our cost of goods is something like 67 cents a patch. We'll come in around there and get that pricing. I do think there's significant potential for first line in kids simply because there is no first line in children. I think there are so many kids, especially young children, ages 6 to 8, who you can't use liquid nitrogen for, even if you wanted to. And so what we do now is reassure the parents and we say, hey, eventually this is going to go away. And, that's not a satisfying experience for anyone, especially, the parent and the child who waited months to come and see you. And so I think the ability to have a prescription product that has, 70 years of use in humans, known safety in children, that has now been reformulated, but with the same, route of administration, I think there's going to be a lot of interest there. And I think the simple ease of use is what will cause this to be adopted as a first-line treatment, even if we cannot achieve, although I think we will, even if we cannot achieve the efficacy of the injectable variant.
Great. Well, Reed, thank you again for being here today. Exciting, again, to hear your updates. And from a pediatrician, this is going to be revolutionary. So glad you're working on it.
We're hopeful. So thank you very much, Katie. And thank you, everyone, for having me today.
All right. So next up, we have Dr. Steve Porter from Risk LD. And to introduce Risk LD, we have Matthew Zenker. Thanks for being here to both of you.
Thank you. And I'll apologize right now. I think my internet is jumping around a little bit, but I'm Matt Zenker, a senior portfolio manager at UH Ventures, the innovation and commercialization arm of university hospitals. And our mandate is to generate alternative sources of revenue for our system. And one of the ways that we do that is build an investment portfolio of promising high impact companies with products that we use and can stand by the results here at UH. Risk LD is a star of our portfolio in that regard, especially as it was conceived, developed, and is being used by some of the greatest minds in labor and delivery, let alone UH. And we are proud to be investors. So it is my privilege to represent UH Ventures and introducing Dr. Steve Porter, an OB hospitalist at UH and CEO of Risk LD.
Thank you so much, Matt. As Matt mentioned, my name is Dr. Steve Porter. I'm a practicing obstetrician gynecologist, and I'm the CEO of Risk LD. So this is the problem. Here in the United States, we have the highest maternal mortality rate of any industrialized nation. Let that sink in for a moment. With all we spend on healthcare, we have the highest maternal mortality rate of any industrialized nation, and it's a problem that's getting worse year after year. And when you break this problem down by the numbers, a couple of very disturbing statistics emerge. The first, we know that the majority, over 60% of pregnancy-related deaths are preventable. And beyond that, there are profound racial disparities in pregnancy health outcomes. Women of color in the United States are 3 to 4 times more likely to die during the course of their pregnancy than their white counterparts. So what's missing? How do we change the paradigm and turn the tide on this appalling maternal health crisis? The answer is technology. At Risk LD, we've developed a software platform that integrates with the inpatient electronic medical record and provides a combination of early alerting and clinical decision support at the point of care on labor and delivery. And it addresses the leading drivers of adverse outcomes in the inpatient obstetric setting. The software does 4 things. It monitors mom and baby continuously. And that monitoring starts from the moment the patient arrives on the labor and delivery unit until the patient is discharged from postpartum. The system diagnoses high-risk conditions in real time. It pushes alerts about the presence of those high-risk conditions. And lastly, it issues real-time and evidence-based clinical decision support for how to manage the condition that's been identified. The clinical decision support of Risk LD is based on a series of clinical practice guidelines that have been exclusively licensed from university hospitals in Cleveland. These clinical practice guidelines, when introduced at university hospitals, were associated with a dramatic 92% reduction in serious safety events. That is the composite measure of adverse maternal and neonatal outcomes. What university hospitals saw is when they introduced these standardized care management protocols, they saw improved outcomes for both moms and babies, and they also saw a 93% reduction in their obstetric litigation costs. The value proposition to our end user is fourfold. With improved outcomes comes decreased costs, decreased litigation costs for the hospital health system that is self-insured, and decreased costs for the payer that is paying for the adverse outcome. There are at least 3 opportunities for hospitals who use our software to realize revenue uplift. The first is by driving increased patient census through a safety branding platform. The second is a revenue optimization play known as critical care billing. And the third is by working with hospitals to increase their compliance with their obstetric value-based care metrics. Now, while there is a core value proposition for hospitals and a commercial opportunity selling directly into hospitals, we are also pursuing a parallel strategy to secure federal government support for our software. And the fact is that the time is now. Not only do we have appalling maternal health outcomes, there is sweeping legislation on the books introduced this year to provide solutions to this national public health crisis. The Black Maternal Health Momnibus was introduced in February of this year, is a sweeping package of 12 different interrelated bills that provide for improving maternal health outcomes and also reducing racial disparities in maternal health. One of those bills, the Tech to Save Moms bill, appropriates $60 million for hospitals who want to implement solutions like ours. We've been working closely since this legislation was introduced with the congressional offices that are leading this work to make sure that the legislative language includes support for provider-facing solutions that are used by clinicians at the point of care. Now, this $60 million includes a $30 million allocation to CMMI, the innovation arm of CMS. And what CMS does is funds demonstration sites for innovative technologies. Those technologies that are deemed to be cost-saving, there is then a mandate to scale those nationally. So this, as a parallel strategy, provides a platform for rapid national scale-up. Our software has been live at all 5 hospitals in the University Hospital system, and we have pilots planned at 5 more, including 2 that are due to launch by the end of this year. We are currently extending the close of our $1 million bridge round in the lead up to our Series A. There is $750,000 remaining in that round. It's worth noting that our software development is complete, and we are a live commercial-grade application that can be downloaded today from Epic's App Store, the App Orchard. So all of the proceeds from this bridge round will be used to support installations at our beta site hospitals. We are led by a team of nationally recognized OBs and supported by a group with more than 30 years of experience in technology development and life sciences entrepreneurship. We are pleased to have the right solution at the right time and to be contributing to turning the tide on this national maternal health crisis. Thank you.
Steve, thank you so much for sharing about RiskLD with us. Again, some really exciting things going on. And I think in my head, this is a no-brainer, right? Why would any hospital say no to bringing this on board? You said you have 5 sites that are live right now. Tell me what the feedback has been from the nurses, the physicians, other clinicians that interface with it.
Absolutely. You know, a few different points on that. From the physician perspective, what I find, because I continue to work as an obstetric hospital on the labor unit that is running our software, is it's a tremendous time saver. Because what it does is it aggregates all the relevant patient information onto a single screen, a single user interface. And that's one of our biggest pain points as labor and delivery providers. We often have 2 systems that we're navigating. We have a fetal monitoring system, and then we have the electronic medical record. Often the fetal monitoring system has all the information about baby, and the electronic medical record has all the information about mom. But within the EMR, the critical information is often housed in multiple tabs. You have to go to one place to learn about the medical history, another place to see what her vital signs are, a third place to see her recent blood work, her lab results, and yet another place to find out what medications she's on. That process is cumbersome and time-consuming, and that is how critical information gets missed. It's what we call the Swiss cheese problem in medicine, a particular problem in inpatient obstetric units, which are dynamic and high acuity settings, and you can't afford for that critical information to get missed. The feedback we've gotten from nurses is that it gives nurses a voice. It gives nurses an objective platform to point to when they are looking to escalate a concern or have a conversation with the physician on the unit. Too often on labor and delivery units, we practice in silos. We have nurses over here and physician or midwife providers over here. Often we are not sharing the complete picture. What nurses find is that there is an objective platform that they can point to if they have concerns about a patient or if they're trying to have a conversation about the progress of a particular patient.
Yeah, that's fantastic. And as you mentioned, the fact that it's based on these clinical guidelines that have already been proven to reduce morbidity and mortality, it seems like a no-brainer to me. So everybody wins, it sounds like. That's exciting. We have a couple of questions from the audience. Folks want to know what your revenue model is.
Sure. So it is a subscription fee where we charge hospitals based on their delivery volumes. So we price it at $100 per patient or $100 per birth, and that provides for that real-time monitoring for the duration of the patient's inpatient admission, all of the alerting, the diagnosis matching, and the clinical decision support. What hospitals also get with that is a reporting package. which is of tremendous value for hospitals. Often hospitals have mandates to report certain obstetric quality measures into state or national perinatal quality care collaboratives. There's not an easy mechanism for extracting that data from the EMR. Often hospitals will have to work directly with their EMR vendors to build customized reporting platforms. And that process is tremendously time consuming and onerous. What our software allows is real-time reporting. So if a hospital wants to find out how it performed on a particular quality metric, like timely treatment of severe range blood pressures, we can issue a report that shows how they were doing last week or how they were doing yesterday. So that subscription of $100 per patient, which is again is volume-based based on the delivery volumes, provides not just the core monitoring, alerting, and decision support, but also the reporting.
That's exciting. And I mean, to your point, the idea that all of this information previously was in multiple systems, probably where you would have to draw that data from, having it in one place, again, makes it much easier for those reporting of outcomes and that type of thing as well, I'm guessing.
That's exactly right. That's exactly right. And also, it's not just reporting into your state or national agencies that need this data, but internally you can mine it as a system if you're trying to drive quality improvement. Because you can figure out how likely are we to adhere to clinical best practices? Are there particular nursing or physician providers that are deviating from those best practices? And that creates opportunities for discussion and for improvement.
Yes, fantastic. One last question, because I'm sure that many of our startup founders and CEOs that are in our audience would be curious to know. Talk a little bit about your experience with the Epic App Orchard, and then how do you plan to move forward with other hospitals that maybe are not up on Epic?
Sure. So we've had a wonderful experience with the Epic App Orchard. When we signed up as a vendor, we were paired with a technical liaison that worked us through the process of getting our software approved as an application. The whole process took about 2 to 3 months, start to finish, but with regular support from that technical liaison. What we're finding going forward is that we have to educate some hospitals that these App Orchard applications are available and that the integration is really a smooth and streamlined process. What we find is that when we approach hospital IT departments, if they haven't had experience with the Epic App Orchard, they get concerned that a software integration like this is going to be a 6 to 12 month process that's going to consume entire teams that they don't have the bandwidth to absorb. Once we educate them about what's required, what has already been done by writing our application to the Epic App Orchard, and really it becomes a 3 to 6 week process of downloading, configuration, and testing, all of a sudden it becomes much more palatable for the hospital IT department. What we're also seeing is that the other major EMR vendors are standing up similar app stores that run on the same technology backbone. So our plan is to make it available on Cerner's App Store next, Allscripts and Meditech to follow. The ultimate goal is to have this software available at any labor and delivery unit in the country, because the stakes are too high and bad outcomes can happen in any possible facilities. The goal is to make this universally accessible.
Well, and with that momnibus legislation that you mentioned, it sounds like that's all possible. So thank you so much for being with us today and telling us all about RiskLD. Exciting stuff happening for you guys. So that's super exciting.
Really appreciate you having us back. Thank you.
Yes. All right. So we are going to move on to our final presenter for the first half of our Best of the Best pitch competition. And I'm going to invite Dan Conley with the New Jersey Angels to the stage to introduce Thomas Barb Porsche from Thrive Bioscience.
Well, thanks, Katie. Can you hear me?
Yep.
Oh, good. So I first invested in Thrive Bio 5 years ago and have twice more invested, including the current convertible note. I'm expecting a 20x plus return because the global platform need of these breakthrough cell assist instruments. And as you learn, Tom Barb Porsche is the best CEO, having enriched many family offices and high net worth angel groups and the like with multiple $1 million exits. So he's a been there, done that. He's on the path. Over 5 years, I've watched pretty carefully, and he keeps delivering what he predicts. Little burp with COVID, but still, all of the elements are coming together, and this now is almost a growth company because people are finally realizing they totally need a cell assist for their researchers to improve, and they can reproduce these stem cell experiments. This is an enormous thing. I'll stop so Tom can go on.
Well, thank you. I just want to make sure you could see my slides. Great, thank you. So my name again is Thomas Farr Porsche, co-founder and CEO of Thrive Bioscience in the Boston area. And we started Thrive because cell culture, even though it's at the center of biomedical research, is still done the way it was 65 years ago. Hours per day are spent manually viewing cells under a microscope by researchers, and little documentation or data is retained. There are extraordinary mislabeling and cross-contamination rates, like 14% to 30%. Billions of dollars are spent studying these variable and poor quality cells with very expensive instruments. And experiments, as a result, are highly variable and non-comparable. There is, as Dan mentioned, a reproducibility crisis. More than 50% of preclinical research is not reproducible. One of our customers, the chair of biochemistry at University of Texas Medical Branch in Galveston, said, "Current tools are failing us because they kill the cells, only give us 2D, single point in time images, and don't capture contextual data or enough data." Our solution are instruments and software that provide advanced imaging, analytics, and automation for reproducible cell, stem, and tissue culture. And that same customer said, "Thrive will change the world. Thrive allows us to see and understand living dynamic cells and tissues in 3 dimensions over time with remarkable images." We have a product family sharing a common database and not requiring FDA clearance. On the bottom left, you see the Cell Assist, which is a $100,000 benchtop imaging instrument. We launched that last year. We're about to launch the Cell Assist 50, which combines the imaging of the Cell Assist with a 50-plate incubator that can image around the clock and remotely, generating enormous amounts of data. And our main focus now is on developing software modules that are really critical in life sciences, such as speeding up viral plaque assays, which are needed for COVID-19, imaging organoids, being able to track a single cell over time. A lot of the benefit of our instrument comes from our really exceptional imaging. We find and track a single cell or a colony over time, even though the plate is taken out and put back in to our instrument, we get within microns of the same cell. We capture up to 60 focal planes, microns apart. So we're able to image in 3 dimensions, including organoids and suspension cells that are floating in the wells. Our typical scan is 10 gigabytes and up to 90 gigabytes. And our sites are generating 2 to 3 terabytes per week, but we make that very simple for them. We hide all the effort that goes behind that with easy to use software. So we produce enormous benefits, not just for lab manager, but also for the researchers. So for a lab manager, we save time and money, we monitor quality, our instruments reduce errors, increases yields. And for the researcher, we accelerate their research. Our instruments, which range from $100,000 to $275,000, have an 18-month payback just on the amount of time it saves from looking for hours under a microscope. And the problem with that is when they put the plate back, all the data is lost. We provide researchers with reproducible, comparable data. And as a result, our sites have had breakthrough insights. So cell culture is an enormous market, $29 billion per year, with the vast majority, 81% of it being pharma, biotech, and cosmetics. We have a serviceable available market of $8 billion, and we project revenues in 6 years of $180 million. And the drivers for that are that we're applicable to all segments of life sciences, and we're expanding our product line, including into bioprocessing, not just research. So like to summarize some key points about Thrive. So we have a number of really significant collaborations, one with a Japanese big pharma, and with 7 leading institutions such as Harvard Stem Cell Institute, Center for Genomics Medicine at MGH. There's limited competition in this market, and so we filed 71 patent applications, of which 23 have issued. And it covers the whole field, even in vitro fertilization, blockchain, machine learning, and for research and bioprocessing. We have a very experienced team. I've started 11 companies. I've had 4 multi-billion-dollar exits. Thrive is very similar to Cytic, where we automated cytology labs, which read pap smears. And that was sold for $6 billion. We've raised $27 million to date. Our largest shareholder is the Estee Lauder family, and we have a number of angel groups. We're conducting a convertible note currently for $2.5 million. We actually have raised $2.5 million, so we're going to increase that a little bit. And if people are interested, we'd like to talk to you. And we target a Series B the beginning of next year, this coming year, for $12.5 million. So I just wanted to thank you and say that we're bringing analytics, automation, and better biology to the life sciences, and we would love to have collaborators and investors help us in this cause. Thank you.
Thomas, thank you so much for telling us about Thrive Bioscience. That's very interesting. And I'm just curious to know, for someone who may not be in the lab or the research space, how about that? We have probably a lot of entrepreneurs and frontline clinicians that have joined us today. Talk about, maybe give us an example or a story of how these tools have led to those really cool, insightful outcomes. Can you share one of those stories with us?
Sure, thank you. And Dan, thank you for your introduction and your support as a shareholder and a mentor. Dan always has 10 or so really wonderful comments on how I can give a presentation better. And we all need to learn and never stop learning. So an example, and the reason that the chairman of the biochemistry department at University of Texas Medical Branch said that Thrive will change the world is because for the first time, they're able to view the effects of viruses on cells without killing the cells, without staining them. And as a result, they don't have to handle the cells again, and they can continue to follow the cells. And further, rather than having to wait 10 days and then staining the cells, using our images, which he called remarkable, they're able to see the effects of the viruses on the cells starting in day 2 rather than in day 10, and to actually collect data about how the virus is affecting the cells. We have 2 customers who are using it for COVID-19 research, and it is a game changer there and in other fields, because we're also able to image organoids. Organoids or 3D cell culture is the fastest growing part of cell culture, because we have 2D culture, which is cells on the bottom of a plate, and we have animals. And when we're doing drug discovery, there's a need for something in between. So we simulate an organ. And the problem with that is they're large. These organoids can be 1 millimeter to 3 millimeters, and no one else can image those the way Thrive does. But because of those 60 focal planes, for the first time, researchers are able to image these organoids. So those are a couple of examples.
Yeah, that's great. Thank you. Very helpful. And I guess one of the questions, though, I still have, because I haven't seen this machine or the output of it, how are you able to look at all of those levels without having kind of the things above and below get in the way? How does that part work?
Yeah. So one word answer: algorithms. And I would say also, very good optics. But one of the reasons why, for instance, other instruments are not able to have imaged viral plaques, the effect of viruses on the cells, is that as the cells die, there's a huge amount of debris. And we're 9 out of 10 engineers here are software engineers and image processing engineers. And so we've developed algorithms and tricks, part of our patent estate, that don't focus on the debris, but focus on the cells. And we have the highest resolution cameras. We use 3 cameras. We use rings of LEDs. We actually model the plate because there actually is a lot of variability and account for changes. We're really focused on cell culture, and we started with cell culture because it is a basis for so much research, as opposed to other companies that started in other fields and are trying to bring their robotics or their microscopes to cell culture. We started with a blank piece of paper to solve these problems.
Yeah, I mean, it sounds like, again, the engineering that you're adding on top of the science clearly is the key here to really taking this to the next level. So that's exciting. We have a question from the audience. Can you talk about your revenue model?
Yeah, so our revenue model is direct sales of instruments. Currently, the range is 100,000 to 275,000. We are developing instruments that bracket those, but our software is going to be sold on a subscription basis. So most of our effort now is on these really needed applications that enhance what this instrument does. So the instrument takes images of organoids and viral plaques and helps with detecting differentiation in stem cells. But we're building toolkits for each of those. And those will be sold on a subscription basis and is really where our margin and our greatest growth is.
Great. And another related question to that, from your presentation, I get you are targeting both researchers and kind of core labs. The comment really was it might be hard for a research lab to write a 200K instrument on an individual grant. So have you seen that, or is it they have the money and they're so excited by the technology that they're willing to spend it on your instrument?
So yes, we see that in a small segment, which is the 20% of the market, which are research institutes, universities, but 80% of our market is biotech, pharma. Cosmetics is one of the largest users of cell-based studies, in vitro toxicology, because they can no longer test in animals. So they've moved to cells, and that's part of the reason why the Estee Lauder family is our largest investor. So for the 80% of the market, there are comparable instruments, many comparable instruments that they already purchase, and we like to obviously point out the greater benefits that they have with ours. So there's nothing unusual about this price range for biotech and pharma and cosmetics.
Good to know. Well, thank you again, Thomas, for being here with us. Really exciting science to hear about. So we appreciate you. And Dan, thanks to you as well.
Thank you, Katie.
So Blake and Elucid has been supported by their early investors, Medtex Ventures, as well as the Global Health Investment Fund. The fund was not able to be here today, but they did want to give me the quote that Elucid is transforming the way we diagnose and treat cardiovascular disease, resulting in improved outcomes and reduced costs by enabling true precision medicine for the first time. So I was very impressed with the imaging, with the modeling capabilities of the Elucid system, and its ability to differentiate between just a flow dynamic versus actual tissue analysis and modeling. So with that, I hand it off to Blake. Elucid, take it away.
So appreciate the time, everyone. I am going to talk you through the problem we're solving with a focus on coronary artery disease, although we do have applications for all of vascular disease. So right now, for the 35 million patients that make their way to non-invasive testing, either because they have high risk factors or are experiencing certain symptoms, currently there's really only two paths that patient will go down. Either they'll be sent to the cath lab for potential revascularization, or they will be put on statins. So of those 35 million patients assessed, about 5 million end up going to the cath lab, where they're looking to assess flow limitation to determine if revascularization is needed, typically stenting. But of those 5 million patients that are sent to the cath lab, under half end up requiring revascularization. meaning for over 2.5 million patients per year, there is an unnecessary trip to the cath lab, an unnecessary invasive diagnostic, radiation exposure to both physicians and patients. And frankly, it's low margin for the facility, and they could be better serving patients that need that care and are also higher margin. Now, with the introduction of FFRCT, you're actually able to make that measurement non-invasively. So you're able to avoid those unnecessary trips to the cath lab, save $4,000 per patient. Fantastic, helpful for everyone involved. But it's missing half of the patient population. While you're understanding those that require revascularization or not, you're missing those patients that have unstable plaque that are at high risk for event, but it's a non-flow limiting lesion, meaning revascularization isn't necessarily the step you'd want to take on that patient's treatment journey, but you're not understanding them, you're not categorizing them appropriately. And then on top of that, you're actually not informing therapeutic treatment for that patient, specifically for them. So we put patients on statins, and we have all these other drugs out there that might be better suited for that patient. What our technology does is takes routine CT angiography, non-invasive tests, runs them through our candid image restoration algorithm, and then uniquely is able to quantify the plaque validated against histopathology. Meaning we have the only FDA clearance to non-invasively quantify tissue as compared to live tissue specimens. And through that specificity, we're able to derive a number of downstream models that inform the physician how to treat that patient individually. The first being an assessment of plaque stability, so whether that plaque is stable, unstable, or stable. The next being that measure of flow in the patient to inform whether or not revascularization is needed and avoid those unnecessary trips to the cath lab. Next, we measure the risk of event over time to inform urgency. And then finally, we'll perform them as the equivalent of a virtual biopsy to actually categorize patients by their disease to inform which therapeutic might be best for them. That being the enabler of true precision medicine in cardiovascular disease for the first time. So, and I won't go into this in as much detail, what we've literally done is taken patients who had a CPA followed by an invasive intervention that removed tissue from the body. We had pathologists review that tissue under a microscope and characterize it. Then we map that back up to the CT angiography. So that is the ground truth for our models, where we can now take in a new CTA and characterize it, not based on Hounsfield units, meaning not based on relative brightness, but looking at patterns and distribution in a manner that emulates what the pathologist would do. And our unique approach with our significant IP and our significant biobank enables us to achieve for the first time characterization validated against histopathology. And this differentiation in our inputs is what enables our differentiated outputs that answer, again, not just who needs to go to the cath lab or not, but provide more information about urgency, about overall risk, about plaque stability, and then about which therapeutic. So we are led by a team with significant experience in the technological and commercial space in cardiovascular disease, as well as significant experience working with the big iron companies. We also have an esteemed scientific advisory board of cardiologists, vascular surgeons, radiologists, and lipidologists who we work closely with in the scientific direction and our clinical studies. Thank you all.
All right, Blake, thanks for going through that. I hope folks were able to hear most of it. We did have you on Pitch Club recently, so what we may do is send out that video and post that video instead so they can get a little bit more of the full pitch, because I think it was at times a little bit hard to hear, but that's okay. Couldn't do anything about it, right?
Yeah, I'm sorry. Unfortunate to hear that, Katie.
Yeah.
Hopefully some of it came through.
For those of you in the audience, we would love any questions that you have for Blake and Elucid to go into the Q&A. But while we're waiting on questions from the audience, I would love to hear a little bit about the precision medicine piece of it. It kind of seems a little bit like a buzzword these days. So what does that look like for your company, and how do you think you're going to achieve that?
Yeah, and unfortunately it has become much of a buzzword in usage, but what we're achieving here, seeking to achieve, is true precision medicine. And I think the best way to think about this is with a comparison to how we treat cancer. So over the past 2 decades, significant amount of work has been done to be able to leverage biopsies and actually run all sorts of omics analyses outside the body to determine, all right, what is the correct treatment pathway for this patient individually? Now, in cardiovascular disease, you cannot go in and biopsy an artery. You potentially cause that very event of 90%. What we've trained our models on is actually that we trained it to perform what a virtual biopsy might be able to, predicting coding and non-coding RNA transfers. And by doing this, our objective is to translate that information into the dominant mechanism of the disease to say, for each patient individually, you might be a likely responder to a PCSK9 versus you who may be a likely responder to a canakinumab versus you who may be a likely responder to a placebo. And these drugs, many of them, are cost prohibitive to prescribe to the general patient population. And because of this, it ends up being that very few end up getting these treatments, even though they may be the best pathway. So by better aligning patient to specific treatment, we're actually able to treat their disease individually, which leads to better outcomes and potentially avoidable events like heart attacks and strokes.
Well, and I would say likely reduction in cost as well, because we do call it the practice of medicine, but you're not having to go through potentially as many trials of therapy till you get to the one that works. So that's exciting.
Exactly. So a drug that might cost $7,000 a year, you can't prescribe that to everyone, but if you prescribe it to those that are going to respond to it, you do actually reduce the overall cost of treatment. It appeals to payers as well and gives them justification for enabling prescription of such drugs that maybe today are too difficult, too many hoops to jump through in order to get.
For sure. Can you talk a little bit more about your business model?
Sure. So we sell licenses with both hospitals and clinics for use of the technology as a diagnostic. We also work directly with pharmaceutical companies and contract research organizations and research groups where we have customized contracts depending on exactly what they're looking to do. So there can be a very, very significant endeavor where we're partnering with them in the very development of their medical molecules to ones that we may be after the fact to help in marketing or to help increase enable another indication that we analyze them on a per-use basis. So with the pharmaceutical companies, it's a bit flexible in terms of what we do on a commercial endeavor. It's really about use.
Great. And one last question. I know you guys are in the midst of analyzing some data, but with your initial trials, do you know how many unwanted cath lab trips have been able to be avoided?
Yeah, so from prospective trials, there's a reduction of about 83% of unnecessary trips, which is hugely beneficial. But on top of that, we've done other trials that are about 30% better at assessing risk of MACE and about 40% better at assessing risk of stroke in the current standard of care. So it is directly quantifiable not only how much money is being reduced, how much cost is being taken out of the system in a way that benefits all, but also these patients that are being sent home unnecessarily, they're able to quantify the difference and say, all right, how many of these patients are moving forward in the treatment process that might otherwise be put on low dose statins and sent home, perhaps incorrectly, they need a more dramatic change in lifestyle or more cancer therapy.
Yeah, this is really exciting technology, Blake. Thank you so much for being here today and sharing about Elucid. We really appreciate it. And we hope to have you back soon to hear more about all the exciting advances you guys are having.
Perfect. Thank you so much. And hopefully with a better working connection.
All right. So next, I'm going to invite Mike back up to the stage, and he is going to introduce Dr. Neil Ray, who is the founder of Radiant Oximetry.
Thank you, Katie. So while virtual conferences have done wonderful things about getting us to be able to put the content forward for you at a short-term notice and collect people from around the world to present, clearly we're running into some technical difficulties on this one, and I'm looking forward to being back in person at some point. I know the rest of you are as well. With that, we have next up Neil Ray from Radiant Oximetry. Now, I first ran into Ray late last year when we was presenting at Pitch Club. AngelMD had actually scoped him out before I even arrived at the firm. And so he's been a long favorite of this company. We've seen him a lot. He's also been supported, as all of these guys have, by other people along the way. In particular, Neil got his step up from the Fogarty Institute, who involved him in an early stage incubation and acceleration trial. And the Fogarty group cannot be here today, but we have a quote from them. Radiant's technology is critical to creating life-changing solutions for moms and babies. I look forward to continuing to follow their success here and overseas. So Neil, take it away, please.
Thank you, Michael. Thank you for your continued support. Can everyone hear me and see my screen?
You're fine.
Okay, great. Hi, everyone. My name is Neil Ray. I'm a board-certified pediatric anesthesiologist turned CEO and founder medical device company called Radiant Oximetry. And we are on a mission to improve outcomes for mothers and babies during childbirth by developing a better fetal monitor. So, reason why we're developing a better fetal monitor is current fetal heart rate monitoring technology looks at changes in the baby's heart rate in relation to uterine contractions. But unfortunately, this technology is over 50 years old with zero innovation. It's difficult to interpret, and it literally has the accuracy of a coin toss in predicting fetal distress. This is characterized by a high false positive and a high false negative rate. So what does that mean? High false positives lead to the overuse of medically unnecessary C-sections because we think the baby's in trouble when they're actually not. This has significant morbidity and mortality to mom and baby. And we have a situation here in the United States where we have the highest maternal mortality of any developed country. The reason for this is complex, but all obstetricians will agree that unnecessary C-sections is a big driver of this phenomenon. In contrast, current fetal heart rate monitoring technology also has a high false negative rate. And this is a situation where a baby's in distress and we fail to identify them. And as you can imagine, this devastates everyone. The child is neurologically impacted, family is devastated, it's very expensive to care for this child, malpractice is high, and we have a newborn birth rate injury here in the United States and across the world that hasn't changed in the past 40 years. So what we're doing at Radiant Oximetry is developing a new type of fetal monitor, one that doesn't look at the baby's heart rate changes, but rather the oxygen saturation. Because you talk to anyone in healthcare, and they'll tell you oxygen saturation is a much better predictor of distress than changes in the heart rate. And what's unique about our technology is we can tell you the baby's oxygen saturation non-invasively directly from the maternal abdomen. We have 8 issued patents to date covering our system, device, and method claims. We also have breakthrough status from the FDA for expedited approval. There are 2 companies currently in obstetrics with breakthrough status. We're fortunate to be one of them, and the FDA has had a very collaborative relationship in terms of getting us expedited to market. We also believe that our preclinical data results are compelling. Current technology has a sensitivity of 85% for detecting fetal distress. We've demonstrated a sensitivity of 99%. Current technology has a specificity of 29% for detecting fetal distress. We've demonstrated a specificity of 95%. What does this mean? If we could improve sensitivity by 10%, we could reduce false negatives by 10% and potentially reduce brain damaged babies by 10%. If we could double specificity, We could reduce false positives by 50% and potentially reduce unnecessary C-sections by 50%. Our vision is to be a disposable sensor company that integrates into existing fetal heart rate monitors. We know that fetal heart rate monitoring technology is a good screening tool, but it's a poor diagnostic. And unfortunately, it's being used as a diagnostic because there's no other diagnostic modalities available. So we want to be the missing diagnostic in labor and delivery. We've surveyed hospital purchasers about the value proposition of our technology and what would they be willing to pay for our disposable sensors. And they've all agreed with the current reimbursement offered in DRG billing systems. They'd be willing to pay $400 per disposable unit. That gives us a 90% gross margin, very attractive business model. We think it's a very, very exciting time in women's healthcare, very much long overdue. Traditionally, only 5% of research dollars have gone into women's healthcare needs, and only 2 to 3% of venture capital has gone into women's healthcare needs. That's changing as we speak. There have been 2 very high-profile exits in the women's healthcare space. Recently, Envision was acquired for $275 million by Boston Scientific, and they raised a total of $17 million to achieve exit. Alydia Health was also recently acquired by Organon for $240 million, and they raised a total of $30 million to achieve exit. We think that our technology is not only attractive to the women's healthcare companies, but also the pulse oximetry companies such as Medtronic and Masimo, as well as the fetal heart rate monitoring companies such as GE and Philips. We've been very fortunate that our value proposition resonates with key stakeholders, and we've been able to raise over $10 million of Series A financing. Just like to take the opportunity to thank AngelMD, Tobin, and Michael for their continued support. We've also earned over $5 million of NIH and NSF grant funding. Our next milestone is to complete a 30-patient pilot study. We'll use that data to raise a Series B of $20 million and use that to complete a 600-patient pivotal study and use that for FDA PMA approval. I would like to add that our technology has the highest regulatory barrier because we're dealing with unborn babies and pregnant women. But we also think that that's a significant advantage. Our 30-patient pilot study is going to be single site and completed at UTMB in Galveston, Texas. Our PI is Dr. George Sadi, who's the editor-in-chief of American Journal of Perinatology. Our protocol has been IRB approved through an NSR determination, and it's a 30-patient study where 20 patients for algorithm calibration and 10 patients for validation. We will report the absolute accuracy, limits of agreement, and sensitivity and specificity. Finally, I'd like to end with some data from Silicon Valley Bank on trends in medical device mergers and acquisitions. And what we can see here is since 2015, the trends have been clear. PMA companies tend to get acquired pre-FDA approval, whereas 510K companies have been getting acquired post-FDA clearance and first post-commercialization. And we think because of these reasons, radiant oximetry is a very attractive opportunity for investment. We have a passionate team dedicated to making childbirth safer across the globe. I'm happy to take any of your questions, and please feel free to reach out to me directly or on LinkedIn if you're interested in learning more about our opportunity.
Neil, thank you so much. Great to have you back, and really exciting to hear some of what's happened since the last time I saw you guys present. Can you tell us a little bit, so for those of you in the audience that haven't heard about radiant oximetry, can you talk a little bit about your preclinical trials that have been done and how you've been able to demonstrate that amazing sensitivity and specificity?
Yeah, so those improvements in our results happened in the last 18 months, actually during COVID when we were sheltered in place. And what we did was we started using a little bit of machine learning to see if it could improve our results, and it did improve them significantly. So we do a pretty complex animal model. It's a pregnant sheep. And what we'll do is under anesthesia, we'll partially deliver the baby, and we'll put lines into the baby, carotid arterial lines, and we'll put an occluder around the umbilical cord. And we'll pack the baby back up in the uterus and pack the mom back up. And then we'll take our non-invasive samples, we'll modulate our umbilical cord occluder to keep blood flow from getting to the baby, and then we'll take direct blood samples from the carotid artery. So it's a pretty wicked, pretty crazy animal model. And we've been able to demonstrate that our technology non-invasively can predict the oxygen saturation confirmed by blood gases.
Wow. And tell me, so your advances, because I agree with you, like those numbers are higher than I saw them before. What can you talk about the changes to was it a change to the sensor themselves that's made the big difference, or what did you guys do?
Machine learning. It was combination of using traditional principles of pulse oximetry and adding in some machine learning, and it's amazing, Katie, how good it is. It's actually kind of scary. Machines one day will be ruling us because they know what they're doing.
Yeah, really exciting though, and congratulations on the progress that you've made. We have a couple questions from the audience, and for those of you that are out there, if you have any more, please put them in the Q&A. Are there patients for which this technology will not work, i.e. body type or skin type or other things like that?
Yeah, that's a great question. We get it asked all the time. And so we can only get enough signal from the fetus when the fetus is less than 4 centimeters deep. So that translates into a body habitus around a BMI of 50. And so we probably will not be able to use our technology on a woman who's morbidly obese with a BMI over 50. Now, here in the United States, where we have an obesity problem, it turns out that pregnant women with a BMI over 50, it's still only 2% of the population. And those women tend to have other coexisting medical diseases that make their delivery high risk. So we still believe that for 98% of laboring women in the United States, that our technology will be applicable.
That makes a lot of sense, for sure. And talk a little bit about, so you talked a little bit in your presentation about your upcoming patient trial. Tell us a little bit about the outcomes that you're looking for related to that.
Yeah, so it's a study. What we do is we look at cord blood gases with the idea that a baby that has enough oxygen during labor and delivery comes out healthy. And the way we define health is they come out with a normal pH. In contrast, a baby that doesn't get enough oxygen during labor and delivery, they come out acidotic. And that's because they'll go from anaerobic metabolism to aerobic metabolism. And in that process, they'll form lactic acid, and then the blood pH will become acidotic. And so looking at cord blood pH is the gold standard for assessing newborn health at birth. And so what we're making the correlation is that a baby with adequate oxygenation comes out healthy, and a baby with low oxygen comes out acidotic.
Yes, that makes total sense to me. I've run those blood gases on those babies, so that makes perfect sense. In your presentation, you talked about the fact that the sensor is just going to plug into existing fetal monitoring machines, which is super exciting. Clearly, those folks there might be an exit opportunity there. Have you had any conversations with those manufacturers?
We have. I'm under non-disclosure agreements, but I can tell you we've talked to all of the key potential acquirers.
I love it. Well, it sounds like the future is bright for you guys, so congratulations. Thank you again for being here today and telling us all about radiant oximetry.
Thank you. I love the work you guys do at AngelMD, so appreciate it.
All right. So we're going to move on to our final company pitch of the day. And I am going to introduce Charlie LaCroix, I'm going to say, from Biologic. And Charlie is going to introduce the next company, which is Bexion. Charlie, if you go ahead and unmute and turn on your camera, we'd love to hear from you. And one thing I am going to say, this presentation is going to be done via video, and then Ray is here to answer questions live. But there will be a slight delay after the introduction for us to get the video up and running.
Well, thank you, Katie. Taking research from Cincinnati Children's Hospital, Bexion has created a drug and technology platform that shows tremendous potential in the fight against cancer. In its earlier years, Bexion was in our incubator space. Since then, and as a small stakeholder, we have followed and continued to support Bexion as it has developed and tested this drug. Along the way, Bexion has developed a great team to move the program forward. So I'm here today to endorse Bexion, and I'm very proud to be able to introduce its founder and CEO, Dr. Ray Takigiku.
So I'm Ray Takigiku, President and CEO of Bexion Pharmaceuticals, and I'm here to talk to you about our company, which is really an oncology-focused company, but have increasingly become a company that is looking at leveraging the untapped mechanisms of the lysosome. BXQ350 is a nanovesicle formulation of sapsin C, which is a normal human protein that can take advantage of three universal attributes of cancer cells. One is an acidic environment, two, exposure of immunomodulatory phosphatidylserine, and three, death pathways mediated by ceramide. In this way, BXQ350 is both tumor targeting and tumor agnostic, having potential to treat a broad range of cancers. Indeed, our phase 1 program has demonstrated a strong safety profile and evidence of activity, encouraging us to be in novel combinations that could significantly improve outcomes. I mentioned death pathways mediated by ceramide, and this figure is meant to show that sapsin C catalyzes synthesis of ceramide across multiple routes, potentially mitigating routes of resistance. We see it in vitro, and we see this happening in our patients. Here's a diagram of what we know about multiple death pathways of tumors with BXQ350. In particular, look at the leftmost panels showing that we have seen similar effects in both a Petri dish and in patient biopsies of a brain tumor. Here are MRIs from a patient with a severe brain tumor known as GBM. He had a recurrence in 2016 when he entered our trial with BXQ350. And to make a long story short, there is no evidence of tumor in that patient today. Here's another patient that had metastatic stage 4 colorectal cancer, and after a year on BXQ, her tumors appeared to go dormant. This is a busy slide, but I wanted to call your attention to the upper left, where the National Cancer Institutes played an important role early in our life. In the upper middle, you'll see the FDA, who have granted us orphan drug designation and eligibility for a rare disease voucher. On the bottom half, you will see we are either in phase 2 or planning to be in multiple phase 2 programs this year onward, looking for positive interim reads and a potential exit in 2023. Some comps are also located in the upper right. On the IP front, you'll see we have applications beyond cancer. So I appreciate every opportunity to talk about BXQ350 and about Bexion. So I thank you for your time and thank you for your attention. And I'm open to any and all questions. Thank you.
Great. Thank you, Ray. Nicely done on the video, by the way. We're excited to have you here. Go ahead and unmute because I would love to ask you some questions. And if you're in the audience and you have questions for Ray or Bexion, we would love for you to put those in the Q&A. But in the meantime, I think there's a lot of science there, right? I mean, there's a lot of science there. And this really seems like a novel approach to cancer treatment. My understanding is that other therapies focus on really genetic mutations or are immunotherapy based. So talk about why this approach is better.
Can you hear me?
Yes, I can hear you now.
Yay.
Perfect.
Yeah, there are kind of three different forms of cancer treatment. I mean, chemotherapy is an old one, obviously, and unfortunately still in wide use. The problem with chemotherapy, obviously, is off-target effects and the toxicities that happen with them. And then there's precision medicine, which, as you said, Katie, are directed more at particular genetic mutations, and often those approaches do work, and that's great. The problem is that tumors mutate then, and then the tumors recur. And then there's immunotherapy, which I love because it does take advantage of our body's own immune systems, which have been developed over billions of years of our evolution, right? And so you would expect that our body would have some pretty great tools to combat disease. And I think BXQ350 is a little bit like that. What we want to do is harness the body's own ability to normalize itself by targeting the lysosome specifically, because the lysosome plays such a critical role in disease progression. And if we can either trick tumor cells into by their lysosomes to killing themselves, or we can possibly revert them to normalcy. That's what we're trying to do.
Yeah, this is really exciting. I mean, it seems like it would be an approach that has less toxicity. Why is that?
So thanks. Yeah, we have seen less toxicity. So we didn't see any, before we could get into the clinic, we had to do some regulated tox studies as part of the IND program. And we didn't really see anything remarkable happening in the rodents and the non-rodents that we looked at. So there weren't any real triggers for toxicity that we were looking for in the clinic. And as it turns out, when we did do our phase 1, we did it both in adults and in pediatric patients, touched almost 100 patients. So 1,100 doses of BXQ, and we didn't reach a dose-limiting toxicity. And we didn't really have any significant adverse events or toxicity showing up. We did have one patient who at one point had an infusion reaction and had to come off study, but otherwise all the other adverse events that were seen were not associated with BXQ. So part of that we think is because BXQ is targeting specifically tumors and not normal healthy tissue. And the other reason I think we're seeing that is because BXQ350 is not overtly cytotoxic. It's more cytostatic. In other words, again, we're trying to manage the disease by kind of nudging these tumor cells into normalcy.
Well, that's very cool because, I mean, as all of us know, cancer drugs all have a lot of toxicity. So exciting results from your phase 1 trials. Can you talk a little bit about, looks like you have several upcoming trials that are planned. Can you tell us a little bit more about those?
Sure. Well, I'm very pleased that we just got a study may proceed for our trial of using BXQ 350 in combination with standard of care for colorectal cancer. It's a chemotherapeutic cocktail called FOLFOX. And the reason we're excited about doing that is that we believe not only will we see synergy without added toxicity, but we may in fact even reduce toxicity. And the reason we think that is because in our phase 1 program, we anecdotally had the surprising but very scientifically grounded observation of some patients experiencing relief from their chemotherapy-induced peripheral neuropathy. So one of the offending agents in this FOLFOX cocktail for colorectal cancer is a drug called oxaliplatin, which is neurotoxic and which does result in a significant proportion of patients getting dose-limiting toxicity from oxaliplatin. And so our hope will be that we'll see BXQ preventing both the acute peripheral neuropathy that's associated with oxaliplatin, as well as hopefully the chronic neuropathy that's associated with that particular therapy. So we're really excited about kind of doing this with the primary goal, obviously, of improving the patient's longevity and overall survival, but also in improving their quality of life significantly.
Yeah, that sounds incredible. That's exciting. Can you tell us, because again, very, very heavily science-based, which of course it has to be, right? At the level that you are right now. How do we know that BXQ 350 only targets cancer cells? And then a follow-up to that, is there a reason to think that it would also be able to get metastases if it is more progressive disease?
Sure. So one of the reasons we believe that BXQ targets tumors specifically is all of the preclinical work that was done by the inventor. Charlie mentioned Cincinnati Children's Hospital as where we licensed this particular technology from. And the inventor was at Cincinnati Children's Hospital, and he published a number of papers where he showed imaging of his research version of BXQ, locating and coalescing only at the tumors themselves. He did also, to your second question about metastases, also observe the research version of the material going to spontaneous metastatic sites. So while we haven't validated that similar sort of data in the clinic, it would obviously be a lot more difficult to do in patients. We did see it across a variety of different tumor models in mice. So yes, I mean, I think there could be very definitely the probability that BXQ, which is administered through IV and therefore systemically delivered, could indeed go to metastatic tumors, even those that might not have been observed prior to therapy.
That's fantastic. Well, Ray, thank you again for being here today. We appreciate you sharing about Bexion Pharmaceuticals with us. And I look forward, I know, to learning much more about the results of your upcoming trials, because this sounds like it could be a game changer.
We're certainly hoping it is, Katie.
Yeah.
Thank you.
Yes. Thanks for being here.
Thanks for the opportunity.
Yes, I'm going to invite Mike up to the stage again to go ahead and bring us home.
Thanks a lot, Katie. Well, for those that were counting, 8 companies. We were trying to have 10 here today. Just proving once again what I was talking about, the virtual versus in-person conferencing. Technical issues along the way have allowed 2 of our brethren to fall, but we will remember them. For those that want more information on the companies that we're supposed to present, we will be sending out their videos and their information on follow-up.
So again, everyone, thank you for joining us. Thank you for your questions. I have to say, I mean, quite a few of these companies, we've seen, some just a few months ago, some earlier this year, and it's really neat to see the level of progress they've all made. So I'm personally excited about that. We do pitch clubs every 2 weeks. It's Tuesday evenings at 6 p.m. Mountain Time, and it's an hour with 3 different presentations in a similar format to what you're seeing today, with presentation and then Q&A. And then we have about a half an hour networking session after for continuing the conversation. And we try to work through all of the different specialties throughout the year, as well as some specific focus areas. You know, we've done digital technology, bioscience, and some other areas. So this upcoming Tuesday, we have radiology. And so we would love any of you who are interested in that to go ahead and join us. And we will be sending out a follow-up email for this event, and we'll provide the link for you to be able to register for that event. And I will also put it in the chat if you want to jump in today. And for the event similar to this one, when you're registered, even if the time for a given week doesn't work out for you, we always send out a follow-up email and access to the video so that you can watch it at your own convenience. So we do hope that you guys will join us. Certainly have loved your questions today.